Okay, so overall you guys did well on the quiz. It was pretty good overall. By Friday or maximum Saturday, I will post all of your grades on Moodle. I still have to grade 1.5 lab reports. Not one personal lab report, 1 lab and a half reports, and the projects. So I'll do that this week, and hopefully you'll get all your grades, including extra credit and everything, before you walk into the final. Final is 200 points, so it might keep you where you are, get you to a higher grade point, or reduce your grade point. So hopefully you'll know before walking into or even studying for the final where you stand. And the review session went well this morning. Hopefully it will go well for Wednesday group as well. The Monday group got this sheet, so I'm going to pass it. If you're in the Wednesday group, grab one and pass. Please, as a reminder, make sure to submit your summary electronically in an email. And I see that there's already a pack of hard copies of the project. Okay. Continuing where we left off, we talked about chemical constituents of concern, residues, and chemical components that can develop during processing or contaminate the food at different stages from production all the way to the consumer. So we just went through a few slides, so it's just introductory again so that we can catch up where we left off and continue with details. So, categories. If I want to categorize the hazards, what would be the main categories? Not specific examples, just categories in general. Remember that pie chart from last time? Yes, Agnes? So we have the chemical, we have chemical, microbiological, mycotoxin is a whole category on its own, and then there was a category of other. So let's name some under chemical. What could they be? Yeah? Pesticides. What else? What do we give the animals sometimes? Antibiotics. What else? Let's name some more. What are some concerns? Yeah. GMOs. What other concerns do we have? Something we add to the food. Example. What do we add to cured meat? Nitrates, nitrite. Sometimes we add a chemical to prevent browning and microbial growth. Sulfites. What could be an adulterant that is not supposed to be in your food and added for gain? Melamine. What could be an example of something toxic generated while processing? Acrylamide. Okay, so there are a whole bunch. We will go over many of them today. What are some quantitative and qualitative methods that we use to quantify them? There are general quantitative and general qualitative. Come on. Yes, Jesse?
Amino acids.
So amino acids can be quantitative, can be semi-quantitative, can be mostly to detect presence or not, so it can be qualitative. Other quantitative methods are mostly chromatography methods, so you do HPLC and GC mostly for all of them, really. There are methods, chromatographic methods for quantification. So let's go specific. But before we go specific, let's talk about handling the samples for analyses. So first of all, we need to get a homogeneous sample. That's like everything. In order to analyze, you need to get a homogeneous sample. And then it's very important that you get representative sample. And this, for contaminants, it's more important than for when you quantify protein content or fat content or so. We talked originally in the sample preparation chapter about mycotoxins and how their presence could be random, not evenly distributed in your sample. So in order to make a conclusion whether your sample is free or does have a mycotoxin contamination, you need to take a lot of samples from different locations. So it's very important to get representative sample of a population, especially when something is dangerous that will wind up and might affect the health of consumers. So this is more critical, this step in this case is more critical than the analytical method that you choose, or the errors that may happen while you are actually running the method. The error of sampling is more dangerous in this case than the error of the analysis. Okay, and then in order to select a method or way of extraction, we have to consider a lot of different characteristics. One, what would be the food matrix? Is my food mostly high in moisture? Is it high in fat, carbohydrate, protein? What's the nature of the matrix and how complex it is for me to achieve appropriate extraction of the contaminant? What do I know about the analyte? Is it nonpolar? Is it polar? Is it charged? Does it like water? Does it like organic solvent? What do I know about it that will help me extract it better? What's available to me in terms of instrumentation? Do I have a GC that I need to does it need derivatization and I have a GC, or I don't have a GC, I have a HPLC and I don't need derivatization? So what's available for me, and then what's my target? Do I want to measure multi-residue, like multiple pesticides in one run, or do I want to measure a specific component and make my method very specific to extract that one particular component and detect that one particular component? Last time we said there are cases where we really need to go with a single residue. When was that? When is it important to get a single residue method? When we are setting what? Limits. When we want to detect or have a, what do you call that? Gosh, I'm losing it. Lowest acceptable value. I can't remember the actual, huh? No, not detection limit. Is what's the least defect? Action level. Thank you. That's what I'm looking for. What would be the concentration above with which we need to have action, basically, or the allowable limits? All right. Okay, so these are the steps which I will explain more in the following slides. So we start with sampling, making sure we follow the proper sampling protocol and proper storage prior to actually running the experiment. Then there will be the sample preparation step, which includes crude extraction and then cleanup, concentration, because sometimes they're present at a very low concentration, so we have to concentrate to allow better sensitivity of our method. And oftentimes we require derivatization if we're using the GC and we want to make sure that it's thermally stable and volatile. Other times you want to derivatize it anyway if we want to measure fluorescence, let's say, or even absorption, and the compound does not fluoresce or absorb light. And then we follow a chromatographic analysis step, either GC or LC. And then we identify and quantify, and then report our results. So here in more detail. So sample preparation, once you select your samples and the representative samples, first of all, you want to remove the edible part from non-edible part. So if you know for sure that the nuts, you're not going to eat the shells, so you remove the shells from the nuts, and then you process the nuts itself. So homogenization follows. Chopping, grinding, blending, you want to reduce that particle size to make sure you have higher surface area. And then you follow extraction based on the solubility or what you know about the analyte. And then you remove the non-solubles, those that you wanted to precipitate out or filter out. And then you do a cleanup, anything that might interfere in the assay, so you do an additional cleanup. Oftentimes it's a chromatography-based cleanup. It could be a size exclusion, where you separate based on size, or it could be adsorptive, whether it is ion exchange or even affinity chromatography, or just hydrophobic or hydrophilic interactions, or it can be very, very specific as affinity. So oftentimes the cleanup involves solid phase extraction. If you remember, you covered that in the GC chapter. So solid phase extraction, you will have an adsorbent basically, or you would have a small column that you would fill with an adsorbent material, and then you pass your sample through. The adsorbent could be an anion exchange or a cation exchange or hydrophobic or hydrophilic matrix. And there's the solid phase micro extraction, where you have a filament, a very thin filament, and you have a thin coating on it with a specific adsorbent, and you immerse it in your solution, or you put it in the head space, and any volatiles can stick to it or adsorb to it. So this would be on a micro scale. This would be just going through a packed column, basically. What is this? The quick, easy, cheap, efficient, rugged, and safe method. So there is a solid phase extraction that has been optimized so that it is quick, it is not complicated, it is cheap, it's not expensive to get the material to do it, and it is safe, obviously. Rugged meaning you have a high sample throughput and a short period of time, basically. So there is a figure in your chapter that gives you an example of that. So a protocol used to extract specific components from a matrix, followed by an analytical chromatography method, followed by detection and analysis. And accelerated solvent extraction, we talked about that in our fat analysis chapter. And basically here you just increase the pressure of your solvent so that you can have a higher boiling point, so you can have a better, more efficient extraction. And then analysis is chromatography based. Quantitative often is GC or HPLC. Qualitative is TLC. Often we need to do derivatization if, like I said, if we need to change the characteristic to suit a particular detection or analytical method. Many of the methods we're going to talk about are linked to MS identification because of the high sensitivity and low threshold and the strong detection capability when using MS. And quite a few qualitative assays using immunoassays, which can be quantitative if we have the appropriate standards. Okay, so now I'm going to go specific to different components. So pesticides. Your current chapter say that there are 500 different registered pesticides. The number grew from 2010 to 2016. It's now 1,300 registered pesticides. And in order to be registered, they have to be the acceptable limits identified for them, and what would be the health concerns. So once all of this has been identified, then they can be registered and then can be used. Often in the past, they were more of a hydrophobic, nonpolar, thermally stable compounds, so they would be analyzed mostly by GC. The most recent ones are polar and non-thermally stable, so they are analyzed using HPLC. So there are the methods that are multi-residue methods, where you can analyze multiple residues in one analysis approach, and then the single residue when you are targeting one. The extraction would be very specific again, and then you can use immunoassays where you have antibodies specific to that one particular pesticide. Or you can, after isolation, that is very specific isolation, you can run GC and HPLC as well. There are semi-quantitative, qualitative methods for screening. When you are detecting presence and you just have a lot of samples and you want to make sure if you have or you don't have a particular contaminant, then you would do thin layer chromatography or enzyme inhibition, which tells you yes, no, do you have sample, do you have a pesticide or you don't. So with the GC, like I said, it's routine for most of the pesticides that are non-polar pesticides. Oftentimes, depending on what the type of pesticides you're looking at, you have to select a particular column and detectors. So the detectors would be based on the components in your pesticide. You have sulfur or phosphorus, you will have halogens, or you have other phosphorus and nitrogen, and oftentimes you actually link to an MS. So the detector is really based on what components you have. And this is just an example where the FPD detector, flame photometer detector, was used. So like I said, HPLC is the method used for polar compounds, not the ones that are not thermally stable. Again, choice of the column depends on the characteristics of the analyte. Oftentimes detection is UV. If you want to use fluorescence, you will conjugate your functional groups on the pesticides with fluorophores so that they fluoresce and you can measure fluorescence. But again, oftentimes detection of pesticides, because of the limits of detections, you have MS linked to it. More often than not, it is MS detection. So with MS, you have confirmation and you can confirm against a library of compounds. So in this case, you have a total ion chromatogram, and then you select for particular ions that you are suspecting, and then when you take one peak and you look at the spectra of the peak, this is done by GC, for example, because you have several fragments. So you will get a compound, and then you can reference it to a reference in your library, and you can have a match. So that would be the compound in your sample, and that would be a reference. So that is an added bonus, and that's why MS is often used for identification and detection. MS is even better, especially when you have a lot of noise and a lot of interfering ions that can be present in one peak. You might have your component of interest and other interfering ions that will have a very crowded spectra. And here's your standard. You cannot really compare this spectra to that spectra. In this case, if you link another MS, so you would take a particular fragment and fragment it more, then you can get a match between a spectra of that one fragment and your standard. So this is again going back to the MS and the benefit of MS in tandem. Okay. We can use pesticides detection using ELISA. And in this case, since pesticides are small molecules, it would be competitive ELISA. So you will have your hydrophobic surface and antibodies attached to it, and then you have your analyte, and then you have a labeled compound similar to your analyte in your analysis, and then they would compete. The labeled one that you've added will compete with the analyte that is actually present. And in this case, the more pesticides you have, the less labeled ones will bind. And the lower the absorption, the higher is the concentration in your sample. So it is inversely proportional to the analyte concentration. Thin layer chromatography, so you can, this is again screening to run multiple samples and detect the presence or absence, not a quantification. So here what you do is you run your samples in each lane, and then you spray the entire layer of silica that you have with an enzyme. The choline esterase is an enzyme common in insects, for example, or pests, and pesticides target the inhibition of that enzyme, and that's how the pesticide is killed, by inhibiting the enzyme that is necessary for that pest. So if the pesticide is present, it's going to inhibit that enzyme. So what you do is you would spray the plate. You will spray it with an enzyme, and then you spray it with a substrate for that enzyme, and you'll get color. Those areas where you don't have color, zones of clearance, that means the enzyme has been inactivated. That means you have pesticides there that inactivated the enzyme. You didn't see a color change when the substrate was added. Okay, mycotoxins. So mycotoxins are basically toxins produced by fungus material, such as Aspergillus is one, Penicillium is another. So there are a series of fungus that can produce mycotoxins. The major toxins are aflatoxins and ochratoxins. Aflatoxin B specifically is very toxic. It's not even, doesn't have an allowable limit. It has to be present at 0 concentration. No tolerance for aflatoxin B. But other mycotoxins, there are set limits for them. So aflatoxins are potent carcinogens. They are immunosuppressive. And ochratoxin is a nephrotoxin that affects the kidney function. So, and they can be present in many different types of food. Nuts would be a common food product that could be contaminated with aflatoxin.
Can you go back to the previous slide? Not this one, the other one. There you go. You said on the bottom, the inactivated enzyme changes color?
No. Inactivated enzyme, you have a clear zone. You don't have a color generated. So when you spray with an enzyme and then spray with a substrate, you get a color. Where you have pesticides, you don't generate the color because the enzyme was inactive. So, other than aflatoxin, do you recall a couple of years ago that there was a recall from Pepin Heights for apple cider? Have you heard about this recall? I think maybe 3 or 4 years ago, there was a huge recall due to presence of a mycotoxin in apple juice and apple cider. Do you know what that? Have you heard of patulin? No? So patulin is a mycotoxin produced by penicillin, not bacteria, fungus. And it is a fungus that commonly can grow on apples if they're bruised, if they fall out of the tree and they bruise. These actually apples that fall out of the tree and they're bruised, they're used for juices and cider, not for eating. So that's why it's very important to monitor limits of mycotoxins in such products. So they had an issue with their detection method, so they quantified and they got less than what actually is in their sample, and they got audited and they found that they had above the limit, so they had to have a major recall for that. Okay, so analysis of mycotoxins, you have reverse phase HPLC, thin layer chromatography, you can do immunoaffinity chromatography, you can do comparative ELISA to detect aflatoxin. So drug residues. So these are your general antibiotics. So they are administered to animals if to treat for certain infection or to treat just at subtherapeutic levels to reduce instances of infection and to increase the rate of weight gain. But there are regulations against having any antibiotics, let's say in milk or in meat products. So the animal before slaughter needs to be off antibiotic for a certain period of time. And also before lactation, they have to be off antibiotic for a certain period of time. Otherwise, these antibiotics will wind up in the milk or meat, and there is a disadvantage for having those. Some people are allergic to antibiotics, so if they are exposed to high levels of them, they will have an allergic reaction. And another disadvantage is you get bacterial resistance over time. With more antibiotics used, you will have bacterial resistance and you won't get rid of the infection anymore. And then major interference is actually while processing the milk, and if you want to ferment the milk or ferment a cheese product or age a cheese for a period of time, then the presence of antibiotics in that will inhibit the fermentation process and the aging process. So we don't want them. So methods of analysis, you have the screening methods. So turbidity measurements, you will have growing or growth media, and you will have in that growth media a sample that you suspect has antibiotic in it, and then you inoculate that media with a particular organism, microorganism, and then you see if the microorganism grow or not. Measure turbidity. If it doesn't grow, you will have lower turbidity. That means you have antibiotics in your sample. The diffusion process, here it's kind of similar principle. You want to see growth, but in this case you have your agar growing media, and then those little dots here are actually small discs that contain your sample. So you put these discs on your media, and then you swap to grow on the media a particular microorganism. In this example is Bacillus stereothermophilus. And here what happens is over time, over the 48 hours of incubation, you'll see growth in certain areas, and if you have antibiotics in your disc, you have zones of clearance where the antibiotic will leach out of the disc and will not allow the bacteria to grow. So you will see that there are zones of clearance where there is no growth. Another screening method is known as charm test. Charm is probably just a name for the company that produced it. So in this case, what you do is you have your sample where you suspect you have antibiotic, and then you have an antibiotic that you suspect present in your sample, but you are adding that is already radio-labeled. And they both, the antibiotic in your sample and the radio-labeled drug, will compete to interact on the surface of the bacteria because they can bind to the surface of the bacteria. So if you have a lot of antibiotics in your sample, then you cannot read the radioactivity because it's competition, so more of the drug in the sample will bind and less of the radioactive or the radio-labeled drug will bind. And the opposite is true. Quantitative is based on reverse phase HPLC at a UV detection, and also you can use GCMS. If you notice, I don't go over principles of HPLC and GC anymore. I expect, will review that, because if the final asks you for principle, you would know this yourself. Okay, so allergens. So you probably heard of the Big 8, right? They call them the Big 8. Why do they call them the Big 8? Do you know where that number comes from? why they're called Big 8? What's so big about them? Huh? It's not, is it just a name? No.
Is that like,
John?
because most people get affected by it?
Yeah, but what puts them in this list? There are a lot of proteins that people are allergic to, like strawberry, huh? No, you raise your hand. He raised his hand, and when I pointed, he said no. What?
Is it just because they're the 8 most common?
You're close. What did you want to say?
They're responsible for like 95% of all allergic.
Well, more like 90%, yes.
90%.
Yes, thank you. Yes, Big 8 are those, the combination of these food sources, but mainly the protein in these food sources that are responsible for 90% of the allergic responses, basically. Obviously, people are allergic to so many different things, but these are the ones that added up together contribute to 90% of the allergy. Reported allergy, that is. But the list might grow to become the Big 9, the Big 10, who knows? The more exposure to proteins from unique foods, the more this is going to be a different list. Okay, so there are concerns about food allergy, obviously, because of the fact that some people are severely allergic and it could lead to actual death. So there is the Food Allergy Labeling and Consumer Protection Act that requires manufacturer to name the ingredients in their original names so that it's known whether or not they contain these allergens. So in the original names, I mean their common names, and also identify source of ingredients derived from any of these 8 allergens. And not just that, actually, if the facility has, like there's M&M, right, but the chocolate M&M, they have to put on the label that they're processed in a facility where peanuts are processed, just for cross-contamination purposes. So it's a really serious deal, labeling. And do you know what the largest food recall up until last year was due to? Allergens. The most recalls in food are due to suspected allergen presence. So it's a lot of loss, economical loss on producers if they mislabel or if they disregard presence of allergen in their product. Okay, so how do we detect them? There are protein methods and there are DNA methods, and you can tell which one is my favorite of the two. So the protein methods, basically it's an immunoassay based methods where you detect the presence of the protein or the antigen in this case by use of antibiotics. So, but what's important or what's is of a concern that we need to be aware of is, in order to identify presence of a particular protein allergen in a food matrix, it's very important to extract it properly, because different proteins require different methods of extraction so that you can get them all the way to the assay. You cannot just use a food matrix as is in the ELISA assay. It has to be extracted. If you use the less optimal extraction method, you end up not detecting that protein. And here's an example. So this is from peanut protein allergy, and then in this case, they extracted the protein using salt in the buffer, and in this case, they extracted the protein without using salt in the buffer. So what you see, that main allergens did not show up here. It's the same sample, but they use different protein extraction method. So it's very important to extract the protein adequately in order to detect it. So ELISA is more quantitative or semi-quantitative, depending whether or not you have standards. Western blot is qualitative. We want to see if you have the allergen or not. Biosensors is basically you immobilize antibodies and you insert them in your sample or extracted sample, and then you get a color change. DNA methods is basically looking at the DNA and looking at the expression of genes that would produce the protein of allergen. And this is done by polymerase chain reaction, which I'm not going to go into details here. But again, there's a concern with the DNA because if you have an ingredient that is, let's say, a protein isolate, and there could be a contamination of that protein isolate that is an allergen in your product, but the protein isolate does not have the DNA. It has been extracted. So if you use DNA methods, then you are probably, in certain cases, not really detecting presence or absence of that allergen. Yeah.
So is the reason you have to be so careful about the protein extraction because you could change the epitopes, like if it was a conformational epitope? Is that why the extraction affects it?
Okay, so that's a very good question and a very protein specific question. So Sonia is asking, do we care about the extraction of the protein because of anything we do that might change the epitope? The answer is yes, but also what I was explaining is something different, because if you want to extract the protein, you want to make sure you use the conditions that will solubilize the protein out of the matrix so that you can detect it. But also, if you heat that protein, you're going to change the conformation. And if you're using kits, ELISA kits with monoclonal antibody, then poof, you're not detecting anything. That's another concern, yes. Okay, here's the wonderful list of other contaminants. Chemical contaminants, oh, the list is very big, and then it keeps growing. So you have all sorts of contaminants and all sorts of reasons for them to be present in the food. So sulfite, like I said, for preservation purposes and reducing browning. Nitrites also for preservation and curing of the meat. Melamine is an illegal adulterant. You have packaging material residues, whether it was from plastic bottles or ink. Furans and acrylamide and benzene are generated during processing. And also monochloropropane is generated during processing, and perchlorate is a chemical contaminant present that from the rocket fuel, or can be contaminating water and leads into food that way. So a long list, we'll go over a few of them. And then there are, obviously some are banned. For example, coumarin, which is a flavor added for flavor, it's banned from certain countries. There are legal limits for some others. There are international contaminants that are not allowed in the food. And some are approved for use, such as monosodium glutamate, and some are present upon processing. So here's the list summarized for you that I have passed around. This is a list that I generated in order to study, and easy to study. So some of these are present in your book already, some are new additions. What they are and why they wind up in your food, why is it of a concern, what cause of health concern it is, where we find it, and what would be the method of analysis. If I were you, I would study this. So it's a nice summary table. So the methods of analyzing these are usually methods that require a lot of work. So it's not that easy. Simple methods mostly are chromatography linked to mass spectrometry, basically, and require certain length of extractions. So it's very important to, some of these methods are still being developed, and it's very important to develop a method with high sensitivity and low threshold, because of the fact that they can be present at a very low concentration, and because of the fact that the concerns are even at a very low concentration, so the limit of having them in the food would be very low. So developing methods that have very low threshold is very important. Otherwise, they will go undetected and cause problems. There are continuous efforts to develop fast methods that have high sensitivity and are inexpensive. So melamine adulteration, you heard the story about pet foods, right? And infant formula with melamine. So basically the manufacturer of the pet food, instead of putting a lot of wanted to replace the wheat gluten and the rice protein, put less of that so that they have economical gain. And they didn't think that they're doing anything bad because they didn't know the health effects of melamine. So melamine, as you can see, has several amine groups, a lot of other nitrogen. So basically, it will be detected as protein if you use Caldal or Dumas, because both are based on nitrogen content. And Caldal is based on the amine content. So they know that they will pass the detection for protein. So they put enough to give you a final protein content based on the specification sheet. So it wound up unnoticed. And that's how it got here in pet food and in infant formula. But melamine is basically a chemical that is used for plastic manufacturing or for mica, for example. So it is not meant for food. And it could lead to several health issues, and also lethalities in infants did occur, and also in pets, unfortunately. But FDA has developed or approved methods to measure melamine in food upon certain extraction protocol using liquid chromatography with triple quadruple MS. So it's MS in tandem, it has a very low detection limit, and also using GCMS has also acceptable detection limit. There are other methods that's been tested or validated for melamine detection, such as IR detection. There are immunoassays, some probes, and electrospray ionization mass spectrometry as well. Sulfites. So there are agents used, like I said, as antimicrobial preservative to reduce browning, but some people have sensitivity to sulfite. That's why they should be stated on the label, and they should be measured to very low concentration or low threshold methods. So there's not one method that can detect all forms of sulfite, but most of the methods detect free form of sulfite and some bound forms of sulfite. A common method is the Monnier-Williams method. It measures the total sulfite in SO2 form that is freed upon the addition of or heat with HCl. So once you heat with HCl, sulfur dioxide is formed, and then nitrogen gas is used to sweep SO2 into a condenser, and using a peroxide solution to oxidize SO2 to H2SO4, and then H2SO4 it gets detected or quantitated gravimetrically or by measuring turbidity. There are other methods. The Ripper method measures free SO2s, and it is based on a titration. And there are enzymatic methods as well, and there are also HPLC coupled with fluorescence detection. Okay, I'm gonna stop here. Everybody's bored, but we'll continue next time.